to our day 5 of Recent Advances in Pediatrics webinar week. I'm glad that all of you are joining us and enjoying our session. And to tell you about today's specialty, it is infections. So to start with, I would like to call upon Dr. Sanjeev Verma, Associate Professor in the Department of Pediatrics and looking after gastroenterology unit. Sir will be speaking about emerging infections. So I welcome you, sir. Over to you.
Thank you, Dr. Arpita. What says host disabled participant screen sharing?
Now you can try, sir. I believe there will be no problem now, sir. You can try.
I hope my screen is visible and I'm audible. My slides are moving.
Yes, sir. Your slides are visible and you are audible.
So I will be discussing on what are the emerging infectious diseases. Before, in last few years, we have seen emergence of certain novel viruses and emerging diseases. In first decade, we have seen SARS, MERS, dengue, even meningococcemia, and in the current decade, we have seen COVID-19 infection along with Ebola and Nipah viruses. So again, we have to think on what are the emerging infectious diseases and which can produce a threat to human beings, especially in our region. So my talk will be on the will comprise of important definitions. What are the recent outbreaks across the world and India? What are the factors responsible for emergence of diseases? What are few epidemiological facts? And I will be taking few of the emerging diseases. To start with, I will again go with the basics of epidemiology, as these are not only important landmarks, but they will also tell us where we are. Is my voice audible? So where we are and what are the steps to be taken for the further prevention of disease progression. Endemic and epidemic we all know, but again we have to go through what is pandemic. Pandemic is an epidemic that is occurring over a wide area and crossing the international boundaries. Mostly it is caused by infectious agents, and this spread of infection is occurring within humans to human. And the most common influenza pandemics have occurred multiple times during the last century and in this century also. There are few stages or phases of pandemic, and WHO has defined each and every phase of pandemic. Phase 1 is basically a virus is circulating within a wildlife, and there is no human contact in this virus to the human contact. Then phase 2, there are few episodes of human infections, but these human infections are very remote, and these human infections are occurring across the world, and they are largely large in numbers, but they do not cause epidemic or pandemics. In phase 3, more than groups of humans are involved, and this stage may have an epidemic. Then coming to phase 4, there is human to human and animal to human virus transmission are causing outbreaks in many parts of the country. In phase 5, this infection has crossed over the one boundary, and at least 2 countries are involved. And in phase 6, many parts of the world or many countries are involved. Then comes the stage of post-peak, where there are possibilities of recurrent events, and then there is a post-pandemic phase, where the disease became pandemic and there may be seasonal levels. So if we can break these chains at phase 2, phase 3, or phase 4, so what are these? That's how we can prevent pandemic. So what are the emerging diseases? Emerging disease is called if the disease is completely a new infection, or it has recently increased in incidence or its impact or severity. It has affected new geographical locations, or it has uh, developed in a new clinical pattern, or developed resistance to existing therapy. So these all are classified for emerging diseases or threats. What are the re-emerging diseases? These diseases are endemic or known for quite period of time, but they are remain present in state and or eradicated. But they have resurfaced and resurfaced in the big way. Example is chikungunya infection in India. Now, emerging infections are made are concerns for humanity, as there are 30 new infectious agents have been detected in last 3 decades. If we see across the world, we have seen in current decade emergence of Ebola virus, novel COVID that we are facing today, MERS COVID, yellow fever, vaccine-derived polio virus, Zika is again a threat, chikungunya. Then in other parts of the world, MERS, measles, typhoid outbreaks have been occurring in the different phases. If we see the Indian spectrum, we have seen Ebola in recent past, Nipah, Zika, chikungunya, avian influenza, and in the last decade, JE, meningococcemia, dengue fever, severe acute respiratory distress syndrome, and cholera. So if we go through the last 70 years of our independence, we can see that we have identified few uncommon viral infections, but we are seeing presence of Nipah virus, Ebola virus, and this Crimean Congo hemorrhagic fever. These are also threat for the or may have may cause emerging infectious diseases because aging outbreaks in the recent future. So in 2019, there is Indian Journal of Medical Research have published an article that showed what are the new possible threats or possible emerging infections. These are basically respiratory viral threats. This could be H1N1, avian flu, H5N1, or Middle East respiratory coronavirus. And then there could be arthropod-borne viral disease like Crimean Congo hemorrhagic fever that has already been detected in Rajasthan and Gujarat. Chikungunya is again with the resurgence and re-emergence. Japanese encephalitis is almost endemic in eastern UP. Usher forest disease, again a viral hemorrhagic disease, mostly encountered in langurs of Karnataka region, and Zika is also identified in mainly southern India. Then arboviral diseases and bat-borne viral diseases like Nipah, Ebola, and COVID infection. So, what are the factors that are contributing to increasing burden of the infectious? Normally, there is enzootic cycle where these wild viruses or emerging viruses make a presence and they maintain cycles within themselves through orthopod vectors, and they keep on these cycles continuing. Accidentally, some domestic animals or domestic animals may get infected and they will cause epizootic disease. And at the same time, these vectors like mosquitoes or orthopod vectors, they can infect humans too. And most of the times, this is the dead end for the incidental. In recent past, what has happened is the ecology of wildlife, human life, and domestic life have changed in a big way. This is because of population growth, human encroachment, ecological manipulation, and on the other hand, in agriculture intensification, then again domestic animals encroachment into the wild area, and there is uh, development of technology, industries, and translocation of human beings. So, urbanization and destruction of natural habitat, climate change, and changing ecosystems, changes in populations of reservoir host or intermediate insect vectors, microbial genetic mutations, international trade and commerce, change in demographic and behavior, lack of public health services and infrastructure, and antibiotic resistance. These all have contributed to emergence of infectious diseases. These are further can be exampled if we have ecological change in form of we have built dams and changes water ecosystem along with deforestation and flood dot have come up. So there is emergence of schistosomiasis, Rift Valley, Argentine hemorrhagic fever, then human demographic and behavioral changes due to social events like population growth, war or civil conflicts, and urban decay. This has led to the introduction of sexually transmitted diseases. International travel has commenced. We are now we can travel from any part of the world to almost other side of the world, and this has led to the rapid transport of one's emerging diseases. And not only this, airport malaria, rat-borne hantavirus infection have disseminated throughout the world. Then coming to the technology and industry, globalization of food supplies, drug causing immunosuppression, they have made emergence of HUS in different part of the world, emergence of transfusion associated hepatitis like hepatitis B, C, and opportunistic infections like Creutzfeldt-Jakob disease. Then there is microbial adaptation and change. This have led to a big way in antibiotic resistant bacteria, and not only antibiotic resistant bacteria, there is antigenic drift within the influenza viruses. Then breakdown in public health measures, and this has led to the resurgence of tuberculosis in United States, cholera in refugee camps in Africa, and resurgence of bacteria in former Soviet Union. So these all factors are contributing to emergence of the new infectious diseases. Apart from this, viral changes do occur with time to time. And what are these? These are mainly antigenic drift and shift. These have come in a big way. What are antigenic drift? These are minor mutations that are occurring because there is no checkpoint within viral mutations, within viral means replication, there is no checkpoint. So accumulation of these mutations may lead to a minor antigenic change, but this will ultimately result in a new viral state. And this occurs frequently. They are easily to treat and mostly occurs in influenza viruses. But once these viruses, they of two origins, they join, their genomes joins, then there is a major shift in there is genomic pattern and there is antigenic characters. So this is a new viral subtype, and this as we have seen in influenza A virus, and this may give rise to pandemics. we can further see a genetic resortment of these entirely new viruses. These will lead to evolve and create new pandemic strain of the viruses. Like in influenza genome, there are 8 loosely linked segments, and these influenza viruses can be there with another virus in the same host cell, and with genetic resortment like human H3N2 flu may combine with bird flu H7N3 and will emerge with H7N2 new influenza A virus. So these shuffles do occur with the viral genes, and this may lead to entirely new viruses. Its antigenic properties are very different. Their approach is very different, and we have seen in the past pandemics due to these assorted viruses. Like 1957, we have seen H2N2 human influenza virus, 1963 H3N2 human influenza virus pandemic, H1N1 in 2009, and 2019 we are seeing COVID-19 viral infections. I'm not going to be in details of new novel coronavirus, but in last 1.5 years we are seeing it or discussing it across the various platforms. But straight away, I am coming to the viral hemorrhagic fevers. Within the viral hemorrhagic fevers, I do see Ebola virus, dengue fever, Yosef forest disease, and Premio Congo hemorrhagic fever that have been observed in recent past in India. But Ebola virus is especially important because it carries a case fatality up to 90%. Though it is originated in sub-Saharan Africa, but its presence have been identified in southern India. So I will take first Ebola virus. Ebola-induced hemorrhagic fever. It's caused by a RNA virus, and this is a encapsulated viral virus, which usually have a transmission within food bats, porcupines, and non-human primates. Incidentally, human gets infected, and these humans can transmit diseases to other humans by direct contact. The average case fatality ratio rate is observed to be around 50% with variability of 25 to 90%. But what we have seen in the recent past, that case fatality have increased from 25% to almost 90%. First time it was observed in the central remote villages of Central Africa, and then in 1976 this has emerged in Sudan and Zaire, and with a case fatality of mortality of 53%. Then Zaire epidemic has a mortality of 59%, and 2014, the last outbreak that has seen, which was the largest and affected the Guinea, Northern Liberia, and Sierra Leone, with a case fatality rate of 90%. How does this transmit? This is transmitted by direct contact, either with blood or body fluids or body tissues. Mostly these infected tissues are eaten by some like dogs or other goats. Then they get infected, and once tissues can get infected. Ebola virus have main target for mononuclear phagocytic system, including dendritic cells, and it severely impairs immunity by viral protein 35 and viral protein 20. Lymphocytes become depleted, and they become apoptosis occurs, and there is inappropriate dendritic cell and T cell interaction. So once Ebola virus infects human being, largely it proliferates in hepatocytes, and later it will cause necrosis of these hepatocytes. These necrotic hepatocytes will ultimately upregulate disseminate DIC, endothelial cell dysfunction, and homeostatic balance. Then Ebola directly invades monocytes and macrophages, and these followed by dendritic cell changes, and this will lead to upregulation of anti-apoptotic gene, and this will cause upregulated cytokine and unregulated storm, and this will again change the coagulation factors such as thrombomodulin and ferritin, and will ultimately again enhance PIC, endothelial cell dysfunction, and homeostatic. This also affects lymphocytes, directly invades. It doesn't enter the lymphocytes, but it will make them almost still, and this will lead to T-cell exhaustion. They do not interact with the dendritic cells, and few markers have been seen that have fatality indicators or hemorrhagic indicators or shock indicators. IL-10 and TNF-alpha are the fatality indicators. Macrophage inducible protein alpha is and interferon inducible protein 10 is hemorrhagic indicator, and nitric oxide and interferon gamma is hypotension and cardiac depression indicator. So, how this clinical manifestation of Ebola occurs? The incubation period ranges from 2 to 21 days, and symptoms include just like any other viral infections like fever, headache, muscle pain, and then diarrhea and GI symptoms and CNS and respiratory symptoms become. Bloody diarrhea and hemochromatosis may occur before the ultimate outcome. In severe cases, fatal outcome is signaled by onset of chest pain along with blindness and bleeding in many cases. We can divide the stages in early febrile phase, last for 3 days, which is usual for any viral infection. From after first 3 days, next 7 days, it's predominate with GI symptoms like epigastric abdominal pain, nausea, vomiting, diarrhea, and this could be associated with persistent fever, headache, conjunctival injection, chest pain, dysphagia, odynophagia, arthralgias, and if we see on the laboratory features, we will find deranged LFTs along with thrombocytopenia, deranged KFTs, and markers of PIC in form of elevated PTAPDT and fibrin split products. Leukocytosis along with thrombocytopenia may be observed. After first week of illness, usually there is setup of shock, and this is manifested by depressed consciousness, rapid pulses, decreased urine output, and this will have elevated lactate and decreased carb. After 10 days, there might be an adverse outcome with the presence of GI hemorrhage, respiratory failure, and other neurological abnormalities. The lab parameter will correspond to the previous findings, and after 7 to 10 days of illness, there might be resolution of GI symptoms with increase in oral intake and decreased fatigue. This may be marker of recovery. Convalescent phase may last for up to 12 months, though there might be presence of arthralgia and myalgias and abdominal pain. The Ebola virus infection can be confirmed by serology or PCR or Ebola. For treatment part, there is no specific antiviral therapy. There is no role of convalescent plasma. Monoclonal antibody cocktails have a definite role as post-exposure prophylaxis, and those patients where hemorrhage and bleeding episodes are there, there is promising role of inhibitors of tissue factor and activated factor 7 complex. Rest is supportive, and we have to titrate fluid therapy because very early setup of fluid overload, as there is compromised function of cardiovascular, cardiac as well as vascular. In last decade, in this decade, we have come up with almost 10 Ebola virus vaccine. Few of them are recommended by FDA or approved by FDA. Mostly have a 2-dose schedule, and if we see there are inactivated vaccine, attenuated vaccine, DNA vaccines, mRNA-based vaccine combined with virus-like vaccine, uh, uh, virus-like particles, or combined with channelized through other viruses. So all of them are giving almost 70-80% to 100% efficacy against fatalities of the Ebola virus. The vaccine are targeted in those areas where there is natural habitat of bat, and there are frequent encounters of human exposures. So all those areas humans are being vaccinated for these Ebola virus vaccines. Coming to the another viral threat, that is chikungunya virus, is that an alpha virus, and this is spread by Aedes aegypti mosquito, also known as tiger mosquito. Chikungunya virus was first emerged in forest of Savannah, and as we have seen emergence of Aedes aegypti transmitted disease within Africa and across the whole of the Southeast Asia, with the increased urbanization, as this is a freshwater mosquito. So we continue to see emergence of these Aedes aegypti related, mosquito related, this vector related diseases. Apart from humans, vertebrates host include sarcopithecus and monkeys and baboons. In almost in 80s, we have seen chikungunya as first time. Then we have seen chikungunya in 2005 with an attack rate of 4 to 45%. Apart from Aedes aegypti, albopictus, and polynesiensis are other commonly involved vectors that can transmit this virus. And not only Aedes aegypti, we have seen a recent epidemic in the Indian Ocean that is caused by Aedes albopictus. We do see frequent mutations in other viruses, and we have seen in this virus also there is mutation occurred in the envelope protein E1 A226D, and this have led to the emergence of this recent epidemic in Indian Ocean. So this is again a viral mutation may again cause emergence of this chikungunya virus in the. It's a positive stranded enveloped RNA virus and endemic in Africa, Middle East, South India, and Southeast Asia. Virus replicates in human cells, including epithelial and endothelial cells, but primarily in fibroblast and macrophages, and it causes cytopathic effects on all mammalian cells. Replication can be suppressed by interferon 1 and 2. So once Aedes aegypti mosquito bites the human, the virus enters the bloodstream or may enter the dermal macrophages, fibroblasts, or lateral cells, and this will further transmit lymph node and will ultimately come to the bloodstream, and this will cause viremia and viremia associated features like fever, malaise, and other. Then it will disseminate lymph node, spleen, muscles, liver, joint, and brain. So we can have all sorts of these type of manifestation with myalgia, joint pain, deranged liver functions, splenomegaly, hepatomegaly, and sometimes it may result as acute encephalitis. Clinical features will comprise of acute onset fever associated with severe arthralgia, And other constitutional symptoms, rash may appear that may last for up to 7 days. Erythrocyclitis and retinitis are the other common ocular manifestation. Diarrhea, vomiting, abdominal pain may occur. Neurological manifestation may include encephalopathy or encephalitis, febrile seizures, and occasionally it may lead to Guillain-Barré syndrome like acute epilepsy paralysis. Other uncommon features are rarely congestive cardiac failure, pneumonia, fulminant hepatitis. We can differentiate chikungunya from dengue with presence of early rash in chikungunya, retroorbital pain in dengue, arthralgia and myalgia are more common in chikungunya. Arthritis is again arthritis and tenosynovitis is again more common in chikungunya, whereas hypotension, profound thrombocytopenia, and minor bleeds are more common in dengue. This is a picture depicting how this chikungunya rash appears. This is the picture of Rocky Mountain spotted fever, where arms and shoulders are more involved with erythematous rash. This is picture of classical picture of blanching erythematous convalescent dengue rash, and this is the picture of this chikungunya virus. Diagnosis: we can isolate virus by inoculations, serological test by doing IgM ELISA, and then we can identify by RT-PCR for chikungunya virus. There is no specific treatment available for chikungunya. Illness is usually self-limiting and resolves with time, but we do need supportive care and rest. Non-aspirin NSAIDs should be indicated for joint pain, and this joint pain can last for up to years, 1 year or 2 years, but unresolved arthritis that is refractory to NSAIDs can be offered chloroquine. Ribavirin has some antiviral activity against chikungunya viruses. For prevention, especially, we have to check mosquito bites and elimination of breeding sites. Elimination of breeding sites is of prime importance with rapid urbanization and dysregulated urbanization. The clear water habitat for mosquito breeding sites are frequently available. So these we have to somehow control these breeding sites. Wearing full sleeves, mosquito repellents, mosquito coils, and electric vapor mats, these can control or repel mosquito bites. Eliminate all source where which can act as artificial containers and which can cause breeding sites for these mosquitoes. Current live vaccine is under trial. Phase 1 and phase 2 trial suggest vaccine is safe and immunogenic, but further trials are still. Coming to another viral threat that has endangered the future of the humans, that is Zika virus. It is again transmitted by Aedes aegypti mosquito, and most of the time it becomes asymptomatic, but 20% of the cases will become stable. This mosquito-borne viral disease is mostly affecting with fever, rash, joint pain, or conjunctivitis, and mostly illness is mild and death is almost rare. Slight fever, headache, conjunctivitis, joint pain, heat rash, muscle pain, these are or general discomfort. These are the main features of this virus. But this virus have of prime importance because of in utero or pregnant women infection, and which have led to this emergence of microcephaly in a big way. And we have seen epidemics in US, Northern America, and not only in Northern America, in Europe, in Africa, and India also. And this will lead to damage to the brain. There might be seizures, there might be CP-like changes. No specific treatment is available for Zika virus, and if the pregnant woman is diagnosed or develops fever with rash, she should be evaluated for Zika virus infection. CLVSG found to be Zika positive. CLVSG for fetal health and anatomy should be seen. Amniocentesis is required for RT-PCR for confirmation of Zika virus infection. As of now, we do not have any vaccine, but mosquito repellent techniques and control strategies are the mainstay. Now coming to the infectious diseases that have emerged in the recent past in a big way. Rickettsial infection comprises a group of organisms which between bacteria and viruses, and they can be divided in spotted fever and typhus. They substantially contribute to the burden of acute febrile illness, and they have seen in the last century, in the earliest last century, the foothills of Himalayas, and they have re-emerged after World War II, and recently have seen acute encephalitis syndrome due to this viral typhus. This is the classification of the rickettsial fever. Spotted fever comprises Indian tick typhus or Rocky Mountain spotted fever that is caused by Rickettsial croni and Rickettsial cachexia. Vector is tick. Then epidemic typhus or murine typhus. Agent is Probojecki and typhi, and these vectors are louse and flea. Then scrub typhus, that is Orientia, Shobumashi. The vector is white. Transitional group is rickettsial fog, that is due to Rickettsia acari, and vector is white. Then others like Q fever, French fever, and radiations. Transmission of these occur through tick, or bite, or flea, or louse. They also act as natural hosts or reservoirs. They maintain infections by transovarial transmission, and main reservoirs are dogs and rodents. Ricketsias are mainly gram-negative, motile, non-spores forming pleomorphic bacteria, and they are obligate intracellular parasites that invade mostly endothelial cells. So these obligate intracellular organisms once invade the human tissue. They mostly localized thermal target cells, especially in fibroblasts, macrophages, thermal dendritic cells, and this is further in the mononuclear phagocytic system, and then they will be released the bloodstream. Then they will disseminate with various organs like liver, spleen, lungs, and brain. Rickettsia is the only organism that goes beyond blood vessel lining and may cause vascular smooth muscles, and particularly arteries. Two main groups, spotted fever group and typhus fever group, can be differentiated. Spotted fever group is mainly transmitted by ticks, while typhus group mainly by body lice and fleas. Typhus group have optimal growth at a little higher temperature at 5 degrees. These all have a almost similar genomic contribution. With the guanine cytosine ratio, they keep on maintaining 32 to 33% in spotted fever group, and typhus group it is going to be 29%. Polymerized actin polymerization of actin occurs in the and they move into nuclei of the host cell and causes spotted fever in humans. But in typhus group, they cannot polymerize actin and mostly found in the cytoplasm of the host cell. Transovarial and transstadial transmissions are essential in spotted fever group, and there is no transovarial transmission occurs in typhus group. I will come to the epidemic typhus, though it is rare in nowadays, but we have seen the epidemics in Russia and Eastern Europe and devastating epidemics during war times. And this is basically human to louse and louse human life cycle. And this human body louse, that is pediculus humanis corporis, is infected for 2 to 3 days. Infection occurred by feeding on infected person, and they excrete rickettsia promogeti in pieces at the time of feeding, and that lies die within 2 weeks. Louse pieces, once rubbed into bite, they cause inhalation and inhalation, sometimes inhalation. They multiply in the endothelial cells of the small blood vessels, and then they will cause vasculitis, they will cause skin rash, DIC, and muscle occlusion. Sometimes there are arterial valves are swollen and necrotic, and they may lead to thrombotic events of the vessels. Incubation period is 7 to 14 days. High fever, chills, headache, cough, myalgia, and this thrombotic event may lead to coma.
Sir, I'm so sorry to
typhus is almost rare. Coming to scrub typhus, that has
Sir, I'm so
in
sorry to
this part of the region in a big way with the emergence of as a contributor of acute encephalitis syndrome.
Dr. Sanjeev, I'm so sorry to disturb. If you could like summarize in 5 minutes, it's already 40 minutes. Thank you, sir.
So, scrub typhus is acute febrile infections caused by Orencia shugumushi. This is again an obligate intracellular gram-negative cocobacilli, non-motile, non-capsulated intracellular parasites, and there is shugumushi triangle that comes across from the Pakistan to Afghanistan. In India, we have seen emergence of this scrub typhus in various parts of the country. And this is the life cycle of the scrub typhus, where mainly it is found in the engorged larva, and when the humans come in contact with these larvae, they may transmit this disease. At the site of inoculation, there is scar formation, then they invade local lymph nodes, and then they come to bloodstream, leading to general symptoms of the intoxication, and then they can invade vascular endothelium, leading to hyperemia system. Mostly incubation period varies, and this may be followed by fever, mental changes, headache, sharp formation, and further dissemination into lymph nodes, hepatosplenomegaly, and this may present with acute encephalitis. This is how Ishar looks like in adults, and this is how rash may appear. We can differentiate dengue with the scrub. Mostly dengue occurs, can present duration of illness before presentation is 2 to 5 days, where in scrub it is variable. Rash is maculopapular that we have seen, and spotted type rashes there in scrub typhus. Ishar is mostly absent in dengue, while tourniquet cyst is present in dengue. Hypotension and shocks presents early, but in scrub typhus it may present in advanced. Mild jaundice are common in scrub typhus. Edema may be present in both the things. Polyserositis is more common in dengue, where pleural effusion along with ascites may occur in dengue. And this, based on these things, SpO2, hemoglobin, WBC counts, SGOT, and serum bilirubin altered and scoring system have been developed and validated. If the score is less than 13, that favors scrub typhus, and if the score is more than 13, it favors dengue. Treatment is basically on doxycycline and the azithromycin as tetracycline and chloroquine being provided in pediatric. Prevention: no vaccines are available. Known infected areas should be avoided. Daily inspection of pits and disinfections of vectors. Post-exposure prophylaxis has no role. and health education we have to show. Now I will skip the rest of the things, but I will come straight away to the take-home message. Respiratory respiratory viruses, arthropod viruses, and bat-borne viral infections and bacterial disease may be upcoming human threats, effective surveillance, diagnostic modalities, and advanced infrastructure is the need of the R and global collaboration of infectious disease is much needed at this point of time. This is my final slide. Any questions?
Thank you so much, sir, for such an excellent presentation. I would request the audience, if they have any queries, they can type in their chat box.
These are my
and sir will be so happy to answer them.